Good day. I'm Max Hegblom, Editor-in-Chief of FEMS Microbiology Ecology, and it's my pleasure to welcome you to today's webinar on microbial ecology for a sustainable future, engaging the next generation of researchers. The mission of FEMS, the Federation of European Microbiological Societies, is connecting, promoting, and supporting microbiology through knowledge exchange and community building for a globally sustainable future. FEMS invests in science. We use the income from our journals to fund activities and support our community, such as this webinar series that enables us to highlight different topics of microbiology and microbial ecology, and allows you to hear from the scientists that are behind the key research published in FEMS journals. Note that when you as an author publish in FEMS journals, and when you as a reader use these publications, you directly support the various activities in which FEMS invests back in science. Our webinar today will focus on our aims to engage and prepare the next generation of researchers in service to the scholarly community, as well as, of course, to the science that they are working on. This was the theme of our recent special issue, Microbial Ecology for a Sustainable Future. With mentoring and support, early career researchers learned editorial skills to ensure a sustainable future for our scientific societies and journals. Indeed, it's the enthusiasm and dedication of all of the microbiologists, the member societies, the volunteers, authors, editors, readers, and staff, and many more is what makes FEMS the organization it is today. So indeed, it's my distinct pleasure to introduce the three editors for this thematic issue who will serve as the host for this webinar. Martina Glodowska from Radboud University, Beatriz Baselga-Cervera from the University of Minnesota, and Damien Finn from the Thunen Institute. They will provide more background on the initiative and introduce the speakers. So I'll leave the floor to you. Welcome.
Yeah, hello everyone, and thanks for organizing this webinar. And yeah, well, I guess now we use this moment to discuss a little bit on our experience as guest editors. So yeah, I don't know, I can only speak from my own perspective and experience, and well, I think it was a great opportunity to get a glimpse on what it is on the other side of the paper publication. It's definitely way more work than I thought, and now I'm way more humble when I'm submitting my papers to any journal, because now I understand what it is to be on the other side. And I think we often forget that it's a pro bono work that we all do for our community, for scientific community. And it takes quite substantial amount of effort. For me, what was the most surprising was to see how difficult it is to get the reviewers. You need to send 20 invitations to get at least 2 or 3. So yeah, I see what's the challenge in there. So that's my perspective.
So I guess I'll jump next. Thank you, Martina, and I echo everything that you say. I would just try to add something new to the conversation. And one of the things that I really appreciated from this opportunity was to have the support. There was always an editorial expert team behind us in which you could ask doubts, because the first time you are an editor and you're trying to navigate what to do, especially if you get inquiry emails, make the decision, it's really nice to have a name that you can go straight with total confidence and say, hey, what do you think about that? This, I'm not completely sure. So that was really fun to do. And I felt really supported by FEMS and the journal in that regard. And also to give also some of the positives, despite it was a daunting task to find reviewers, when you find some of them, then it was an opening conversation for collaborations that I was not envisioning. So some of the reviewers came back to me, and now they were interested in my research, and it have trickled down in pathways I was not expecting. So it was a great opportunity as an early early career researcher. And I guess I'll pass it to Damian if he wants to add something or his perspective.
I would definitely add a big thank you to Sadie. You were fantastic. So much help. Yeah, I just kind of moving on from what Max introduced at the very beginning is that my thoughts have always been that FEMS is this sort of like fantastic community. And I definitely profited off that very early on in my career because I started in Australia and then like FEMS in Genève was my first actual big conference and getting involved in things. And I've been a very happy recipient of some of the research and like traineeship grants that FEMS provides, because that's a great sort of thing that lets you sort of move around and explore other labs in Europe and stuff like that. And now a bit like Eurovision, I guess. I'm an Australian who's stuck here in Europe and no one's told me to leave yet, so that's great. I'm just going to sort of keep a low profile and just kind of enjoy it here while we still go on. So yeah, my suggestions would be to everyone, take advantage of everything that FEMS offers because there's so much there to do. And then it would be nice if we ask you as like a publisher in the FEMS journals, if we're like, hey, can you like be a reviewer for this one little thing that's absolutely in your field? You're like, yes, of course, because I am also part of the FEMS community.
And I think one aspect just to add, no good deed goes unpunished. So when you say yes to a review, well, then suddenly you'll get a second request and who knows it, you'll become a guest editor, an editor, editorial board member, or so on. So again, but I think it's really this aspect, it's an opportunity to give back to the community, which I think is actually quite fun.
All right, so is it time to move toward our presentations? So it's my pleasure to introduce our first speaker, who is Jessica Bennett. Well, I had the privilege working with Jessica in the Department of Microbiology at Radboud University in the Netherlands for past 4 years. Jessica has been, since the very beginning of her academic career, linked with the methane cycling. She started very early in her bachelor thesis to work on this topic, and she continued until today. She's an aquatic microbiologist and recently completed her PhD thesis and is now very open for new challenges. So if you have any exciting postdoc opportunities, don't hesitate to shoot her an email. She's looking for something a little bit toward more application of what she's been doing. And yeah, I will be happy now to see Jessica's presentation. And yeah, the floor is yours. So if you can share your presentation with us. I'm going to turn off my camera and the floor is yours.
Cool. Thanks, Martina, for introducing me. I'm very excited to talk today at the FEMS webinar about the seasonal dynamics of microbial methane removal in the water column of a coastal basin, which was actually part of my PhD thesis. And I want to say at this point that this was a collaboration within the Marix Synergy ERC project led by Caroline Slomp and Mike Yetten, which aimed to unravel the mechanisms behind methane and ammonium removal in redox transition zones, specifically in coastal ecosystems. And as you can see in this nice family tree that my colleague Olga has made, in yellow you see all the biogeochemists, and in red you see all the microbiologists. So it was really a collaborative team effort where each of us looked at different aspects of methane or ammonium removal. And now, why is it important to look at methane removal in coastal ecosystems? You all know methane is a potent greenhouse gas, and I will spare you the terrifying hockey stick graph from the ICCP report. But I do want to show you that the coastal ecosystems are actually biogeochemical hotspots for methane emissions. And even though they are only a small area of the global surface ocean, it is estimated that they contribute up to 75% of total marine methane fluxes to the atmosphere. And these fluxes are likely to increase in the future because of ongoing eutrophication and the co-occurring increased stratification due to warming and the expansion of hypoxic conditions. However, these predictions still come with very high uncertainties, because on one point, these systems have a very high spatial and temporal variation in their biogeochemistry. And linked to that, there's still a lack of understanding of how this variation actually influences the microbial activity and ecology of the microbes behind methane cycling. And so, because in coastal ecosystems, methane emissions are actually the result of methane production and microbial methane consumption. So methane is produced as the last step in organic matter degradation by methanorganic archaea, and then methane travels upwards through the sediment, where it can luckily be subsequently oxidized by a consortium of methanotrophic microorganisms. While in marine ecosystems or in marine sediments, this methane is filtered out in the sediments quite efficiently. It has been shown that in coastal systems with a very high sedimentation rate and input of organic matter, there is an imbalance between methane production and methane consumption in the sediment, which leads to very high methane fluxes into the water column. Luckily, in the water column, aerobic methanotrophic bacteria can also further reduce methane emissions. And it has been shown that these are actually very important, especially for shallow marine waters. However, it is still unknown how the seasonal dynamics of shallow marine waters influence this methane biofilter, because the water column chemistry can change drastically with the seasons. So we have to look into this as well. For instance, in spring, the water column is generally well oxygenated and mixed, and there is good oxygen transfer from the atmosphere into the bottom water. However, when the temperature increases, the surface water warms faster than the bottom water, and we get a density difference, which leads to that we have during summer stratification a warm, low density top layer that is distinguished from the cold, high density bottom layer. And the transition between these two layers form a barrier for vertical mixing, as well as diffusive transport of, for instance, oxygen. So when we have this stratification, oxygen cannot or diffuses very slower into the cold bottom water layer, and together with oxygen consumption, this can lead to anoxia. This is called an oxycline, and then this can lead to anoxia in the bottom water. And then when the temperatures are lower again and the winds are stronger, we have a mixing of the water column again in autumn, which replenishes the bottom water oxygen concentrations again. So as you can see, there's quite a lot of changes during the season, and how are the microbes affected by this. A very good study site that we looked into a lot is the marine lake Grevelinge, which is an estuary and is now brackish water. And it suffers from high eutrophication and reoccurring anoxia in the summer months. And we focused specifically in the quite deep part, well, at least for Dutch standards, it's quite deep. Because in this deepest part, we did see that there is a lot of organic matter input, which disbalances this methane filter in the sediment, and we have a lot of methane coming into the water. So in 2020, we already looked at the situation in the water column during summer stratification, and we saw that we had this nice counter gradient of methane and oxygen. So methane is the dots and oxygen is only the line. And in this methane oxygen counter gradient, the methanotrophic bacteria, which you can see here on the right as the gene copies of 16S rRNA sequencing, we populated this methane oxygen counter gradient and build this methane filter. And together with metagenomic analysis, we could see that the genomic potential shows a high adaptation to low oxygen concentrations, which means that there's probably high resilience of this methane filter towards changes in oxygen concentrations. Because this is, and as I showed you before, this is important, and also in our study site. Here you can see the seasonal distribution of oxygen concentrations. So the y-axis is the depth, and the x-axis are the months. And we see a lot of oxygen in spring. Then we have stratification with anoxia in summer, and then we have again reoxygenation in spring. So we wanted to know, how does the seasonal dynamics affect the methane distribution in this system? What effect does it have on the microbial methanotrophic community structure? How does it affect potential for methane removal? And also, how is this reflected in the water-air fluxes? And we did this by a very extensive seasonal sampling in 2021 with the research vessel Navicula from the NEOS Institute. And at this point, shout out to the crew members and also to my colleagues. We were a very enthusiastic, hardworking crew, which led to me being a very happy PhD candidate, because we had a really big data set and we could take a lot of samples. So we took samples for water column chemistry. We also took samples for DNA extractions and analyzed it by 16S amplicon sequencing. We also did a bunch of potential methane oxidation rate measurements. And we also measured in situ methane fluxes with a floating chamber connected to an online Leica gas analyzer. And now I will walk you through our most important findings. So first of all, we start with spring again. In March, the water column is mixed and oxygenated. There's very little methane, which you can see as the red dots. And then the microbial community was very underwhelming. You see maybe a little bit of the yellow color, but in fact, still the community was actually the same as in doing summer stratification the previous year that I showed you before. And then by July, you can see in blue the oxycline. From 15 meters onwards, there was no oxygen anymore. And we see that there's a lot of methane coming from the bottom water into the, from the sediment into the water. And very impressively, the methanotrophic community increased up to 10% of relative abundance by July. And now we also looked into the capacity of this methanotrophic bacteria to oxidize methane aerobically. And we could see that methane oxidation could be activated in all our incubations pretty fast, actually. And the rates were also quite high. However, it was interesting to see that the methane oxidation was most active in the anoxic part below the oxycline, as opposed to in 2020. And also the oxycline and the methane gradient were decoupled from each other. So this is interesting, because how do then aerobic methanotrophs oxidize this methane? It has been shown in other studies that methanotrophic bacteria can oxidize methane with alternative electron acceptors. And in our study site, we think that it could have been potentially coupled to iron oxides, or also an external electron transfer via dissolved organic matter. But also we cannot exclude cryptic oxygen cycling, which has been a hot topic in the recent year as well. So now we see that we have an active methane filter from the water column profile, and we could also see this actually in the fluxes. So here you see the fluxes of methane throughout the year. And we see that in the summer months, the fluxes are actually lower. However, I want to point out that the fluxes are still quite high. And also, I want to mention here that my colleague Olga also published a nice paper from the same year, where she shows that actually bubbles coming from the sediment can directly escape to the atmosphere, so they can surpass the methane biofilter. So even though we have a working biofilter and it's removing already quite a lot of methane, there's still a lot of methane coming out there. And yeah. And also, moreover, we also see that with prolonged or the longer in the summer, the higher the methane fluxes are. So I added here view August, September as well, and then also the fluxes. And yeah, we see this, and we so something must have affected the activity of the filter, and we think that it might have been something to do with a weakening of the stratification. So potentially there was increased turbulent flux, which could have made the time where methane can be oxidized in the water column shorter, so more methane can escape. We also saw from the nutrient profiles that there was a accumulation of nitrite, which could have inhibited methane oxidation activity. And connected to this, we also think that a general shift in the microbial community composition through succession or also other events could have affected the interactions between methanotrophs and the rest of the microbial community. And I want to just say that this is a controversial effect by a mixing event. It's always like there's papers that say mixing will release methane. Other papers say mixing has a positive effect on methane removal because there's more oxygen and the methanotrophs can oxidize more methane. So as a summary, made some take-home messages for you. So the water column methane filter is indeed very important in these eutrophic coastal waters because it's removing a lot of methane when it counteracts the high diffuse fluxes from the sediment. Still, the methane emissions are pretty high and they are variable throughout the year. And we also see that the methanotrophic community is strongly influenced by the seasonal dynamics, and also the functioning of the filter is influenced by this, which is connected to the succession of the MOB community with the water chemistry. But also we showed that oxygen availability really does not determine the functioning of the microbial methane filter, because we see that there is high potential for anaerobic removal, and also there is high potential for a negative effect for a mixing event and potential reoxygenation. So it's not as easy to determine this. And yeah, as a last statement, it is indeed a complex network of factors that drive this methane, microbial methane filter, that has a strong interaction between water chemistry and microbial community structure. And I think this shows how important it is also to incorporate these questions into interdisciplinary work, because yeah, it's more complex than it seems. And with that, I want to thank you for your attention. And yeah, you can also contact me via LinkedIn or send me an email anytime. I'm looking forward to the discussion.
All right, thanks, Jessica. And yeah, indeed, we forgot to mention that the question session is going to happen in the end of all three presentations. So just put your question in the Q&A chat, and then Max will be picking up some questions, and we are going to steer the conversation later on. And with that, I think it's our second speaker that will be introduced by Bea.
Hi, thank you, Martina. I have the privilege to introduce Juan Carlos Cambronero Heinrich, which is currently a postdoctoral researcher for the University of Padova in Italy. And he's going to be moving to his second postdoctoral position in New Mexico State University in the United States, in which he's going to focus in the transmission of vector-borne disease in Costa Rica. Just to highlight some of the really important work that Juan Carlos is making in microbial ecology and entomology, I want to highlight that he recently received the honor of a graduate student award of the Entomology Society of America. So with further ado, I'm going to pass it on to Juan Carlos, in which he's going to present the defensive and nutritional roles of Erwiniaceae bacteria in ambrosia beetles. So the floor is yours, Juan Carlos.
Thank you, Bea. Thank you for this nice introduction, and also would like to thank FEMS for the invitation to present in this webinar. So I will start sharing my screen. As Bea mentioned, I will talk about Erwiniaceae bacteria and the role in ambrosia beetles. I'm a postdoctoral researcher and a PhD from the University of Padova in Italy. And in general, I will start commenting on ambrosia beetles because I'm sure many of the people in the audience don't know what they are. Ambrosia beetles are a group of weevils in the subfamilies Scolitinae and Platypodinae, and they are mostly distributed in tropical and subtropical regions of the world. But in recent years, they have been gaining scientific interest, and it's mainly because there are some of these insects that are high successful invaders. And factors such as globalization and the transport of wood materials have eased the colonization of new environments by these insects. Ambrosia beetles are wood botters. That means that they live inside wood, and they excavate tunnels deep into the xylem of trees, where they cultivate fungi. The defining characteristic of ambrosia beetles is that they are associated in mutualistic relationships with fungi that they harvest in these tunnels that they excavate. Many species are polyphagous, and this is quite important. That means that many species can attack and colonize multiple species of deciduous trees mainly. And this is also important related to their role as pest insects, because many of them are able to affect different crops or orchards, for example, apples, avocados, and so on. And many species are very destructive, actually. Some of them are very important in countries like the USA, in areas in Europe, Israel, for example, for the production of avocado or apples, for example. And another characteristic that is very important is that they show high social levels of behavior. And this is interesting in general because they are defined as fungal farmers. They are well known because of their interaction with fungi, and they are usually an book example of this characteristic. I will talk about their interaction with microorganisms in general, but I will focus in bacteria. And I will introduce the topic before I go to the results in the manuscript we publish. In general, when people study ambrosia beetles, they usually focus in the microbiology in the interaction of ambrosia beetles with their fungi. These mutualists they possess, or they transport from plant to plant. And this might be for many reasons, but one of them it's because actually when we dissect a tree that is infested with ambrosia beetles, you can actually see with your naked eye the growth of fungi. But for many years, the presence of bacteria there has been largely unexplored. Actually, we know that bacteria are ubiquitous, but in total, to date, there's only 8 metabarcoding studies describing the community of bacteria associated with ambrosia beetles. And these studies actually encompass only 13 species of ambrosia beetles in the tribe Silobrini, mainly. And to give some context, we will see that there's at least 3,000 species of Scolitine beetles that are described as ambrosia beetles. So there's very little we know about the bacterial community associated to ambrosia beetles. What we know from the papers that are published on this topic is that the community of bacteria associated to ambrosia beetles is highly species specific. So, for example, in a community or in an environment composed for populations of different ambrosia beetles, native and exotic, you will notice that the bacterial community associated to native and exotic species, even in the same environment, are very different. But that doesn't mean that some of the microorganisms associated to them are actually probably associated in a long period of evolution and share between the different groups. There's also some differences related to the environment that we study. For example, if you study the wall insect, you will notice that the community is kind of similar to the community in the garden. They are usually sharing microorganisms between the community in the beetle and the garden, but the abundances are different. So probably because of the activity of harvesting or fungal farming that the beetle has, the community is kind of shaped by this in the fungal garden, and also because, well, the fungal garden has to adapt to the plant environment. But the main point to highlight in what we know about the community of bacteria and ambrosia beetles is that we don't know a lot about the function of the microorganisms associated to them. In general, when people study these communities, usually we have very descriptive metabarcoding studies that try to assume possible functions from what we know from other insects. For example, bark beetles that are a group of scolitids that is more studied. So there's a need to study the function of these microorganisms, mainly because they can be probably a third part of this fungal insect interaction or this symbiosis, the ambrosia symbiosis, and that they might be very important. For example, when we study one of the most commonly studied ambrosia beetle, Silobrinus saxeseni, a native of Europe, you will notice that the community of the beetle is kind of diverse and dominated by the gram-positives and negatives. We can see Pseudomonas, Bacillus, and Streptomyces being very important. But when you try to focus on the function of these microorganisms, you will notice that there's only one study assessing the function of Streptomyces in Silobrinus saxeseni. The rest of what we know about function is related to assumption from other insects on similar to ambrosia beetles. And when we study the fungal garden, you will notice that taxa of Pseudomonadota are very important. And these taxa are actually also present in the beetle. And I will focus on Erwinia, the bacteria I will start comment on related to the paper I published in FEMS Microbiology Ecology. But the most abundant bacteria, Pseudoxanthomonas, is a bacteria that has not been studied, and we don't know exactly what it's doing in this environment. So the big question is, what are the functional roles of these abundant bacteria? And actually, what we did was something very straightforward. We use culture-dependent methods to study the function of this abundant bacteria, this Erwinia. In fact, we have a big experiment in the field where I obtain samples from different species of ambrosia beetles, and I did bacterial collection and fungal collection from these associated microorganisms. And after barcoding, I noticed I obtained some Erwinias from Silosandrus crassisculus and Germanus and Saxeseni that kind of were fitting with this Erwinia from the fungal gardens that has been reported in metabarcoding studies. So we decided to study more in detail these bacteria. First, we did a phylogenetical analysis, and we noticed when we did the barcoding study that the 16S, when using a phylogenetic using the 16S RNA sequence, that there's a kind of a cluster of this Erwinia associated to ambrosia beetles. Actually, all of the isolates reported in the gene bank, and the sequences reported in the gene bank for this cluster are associated to scolitids. Scolitids including ambrosia beetles like Silobronidus saxeseni and Silosandrus species, and other bark beetles like Ips typographus, that is very important in Europe, and then Doctonus species that are bark beetles that are quite destructive too. So we kind of make the hypothesis that probably this cluster of Erwinias is kind of important and associated to ambrosia beetles. And we made the hypothesis that probably they have been evolving together, and that this lineage is isolated from other lineages in Erwinia. And is also something very important because, as you will know, there's many species in the family Erwiniaceae that can be plant pathogens, and we didn't want to just assume that this Erwinia is just a plant pathogen associated to ambrosia beetles. So with that in mind, we have the hypothesis that this Erwinia was a facultative symbiont associated to ambrosia beetles in general, and probably to other scolitids. And we decided to target possible functions sequencing the genome of the isolate of Erwinia associated to Silosandrus crassisculus. We obtained a big genome of 5.4 megabases. We target different functions that might be associated to a facultative symbiont. And first, we went to nutritional benefits of the bacteria, and we found that the bacteria is able to produce essential amino acids. Styrina and threonine completely, they have the full pathways to produce these amino acids. And they have also the capacity to produce enzymes for the removal of plant polysaccharides, cellulasas, for example. And this is kind of important, and because these bacteria living in the fungal garden of ambrosia beetles might be helping the fungi and also the beetles to modify the plant environment and to also to provision nutrients to the ambrosia symbiosis. Then we focus in another type of benefit, and we thought about defensive symbiosis. Defensive symbiosis is when microorganisms produce an effect to protect against competitors or pathogens or parasites. And we look for clusters of antibiotics. We found clusters of antibiotics that are kind of not fully descripted in Erwinia or in this isolate that we have. So we decided just to try to test the hypothesis of a defensive symbiosis and try to see if these clusters might be kind of relevant for the ambrosia symbiosis. So we did something very simple, also cultural dependent methods. And what we did was to expose the Erwinia isolated from Silosandrus crassisculus in interaction assay to fungi from different groups, actually. So we decided to put an inocula of a certain concentration of the Erwinia in the middle of the Petri dish. And you will see in the right a petri dish that we put also 2 inocula of fungi of a certain growth time. In the left, you have the control, a petri dish with only the fungi. And after a certain time of growth, we compare the growth of both petri dishes to see the effect of the Erwinia in the growth of the fungi. The first group of fungi that we exposed to Erwinia was a nutritional symbionts of ambrosia beetles. As I commented before, ambrosia beetles are associated to fungi that are their main mutualist. And in a scenario of a defensive symbiosis, Erwinia shouldn't have any effect in the growth of, or at least not a negative effect in the growth of nutritional symbionts of ambrosia beetles. And that was in fact the case. When we compare the growth in the control groups and in the experimental groups, we notice, and you will see in the graphs down that quantifies the effect, that there's no significant effect in the difference of in the growth. That you can see that as an NS in the figure. So this is kind of consistent with the defensive symbiosis, but we need also the certain effect in other groups of fungi to kind of support the defensive symbiosis. So we try to test the effect on other fungal associated ambrosia beetles, fungi that usually interact with ambrosia beetles in the environment that is the plant. And we did the ECI with endophytes that are usually, in some cases, described as plant pathogens. And we noticed that in some cases, some plant pathogens or endophytes are inhibited by the Erwinia. And you can see that in the Plodia seriata that you will see in the petri dish to the right, an halo of inhibition. But not every fungi shows the same effect. So for certain fungi, there's an effect of Erwinia of inhibition. And then we also test this effect on mycopathogens and entomopathogens. Mycopathogens, we have certain species of trichoderma that are usually naturally associated to ambrosia beetles. And entomopathogens, we have different fungi, for example, Bouveria baessiana, that is usually naturally occurring in the fungal galleries of ambrosia beetles. We notice that certain species of trichoderma are inhibited by the bacteria, not all of them. Certain entomopathogens too, for example, Bouveria baessiana is inhibited. So this is kind of consistent with the offensive symbiosis hypothesis. And this is also very important in terms of biological control, because in some cases people or scientists or practitioners try to control insects with entomopathogens, and the truth is that ambrosia beetles might be protected for some of them thanks to the association with this bacteria. And also mycopathogens, trichoderma, are well used, and you will notice that trichoderma is a parasite of the fungal gardens of ambrosia beetles, and some species are also inhibited by this bacteria. So with this in mind, we concluded that this Erwinia, isolated from other lineages in the Erwiniaceae family, are probably widespread in ambrosia beetles and Escolitids, but the bacteria has a nutritional role in ambrosia beetles in providing essential amino acids, probably, and synthesizing enzymes for the removal of plant polysaccharides. And also that we can kind of support the hypothesis of this bacteria as acting as a defensive symbiont, a facultative defensive symbiont. And this is my presentation. This was published in 2023 in FEMS Microbiology Ecology. And I would like to thank University of Padova and Fondazione Casa di Risparmio di Padova e Rovigo for the scholarship that allowed my PhD studies. My supervisors from University of Padua, Andrea Battisti, Davide Rosati, and from University of Freiburg in Germany, Peter Biedermann, and other colleagues like Giacomo Cavoletto and Giacomo Santoyema that were always present in my projects. Muchas gracias, and that's it for now. Thank you.
Just a quick reminder, please type your questions in the chat. We're gonna have a Q&A at the end of the 3 talks. Amazing work, as I previously said, and I'm really excited to see your questions. I'm gonna pass it to the next speaker that is gonna be introduced by Damian.
Thank you, Bea. I would like to introduce our third speaker for the day, which is Dr. Priscilla Carrillo Barragan. I hope I get that right. I can never roll the R's. She is based at the University of Basel. And in our thematic issue on sustainability, Priscilla's paper is titled Clean Your Own House First, Integrating Sustainability into Microbiology Labs. And I feel like this paper, this perspective piece, really drove to the heart of the whole thematic issue itself, which was about its sustainability, of course. And it's, looking at how we, and not like the abstract we, like as a society, but we as in you and me in the lab every day, how we can improve what we do to reduce our environmental footprint and just do our own little part to make the world a little better place, because there's so much plastic waste. Oh my God, so much plastic waste. Anyway, when the paper was first released, the first thing I did was share it around our lab, and we all had a very good read of it, and I recommend you read it too. Okay, so please, Priscilla, the floor is yours, and yes, show us how we can all be better people on a daily basis.
Thank you very much for that exciting introduction, Damian, and I will be happy to now share my screen and my presentation with you. It is, I have to tell you, it is quite different from the very interesting talks we had before, so bear with me and we'll get to the publication eventually. My presentation, Integrating Sustainability into Microbiology Labs. My name is Priscilla Carrillo Barragan, and now I am no longer based in the University of Basel, but that just happened recently, so I am in the University of Zurich now, but I published this perspective while I was in Basel, just that. But just to like, as an icebreaker, let me talk about myself. I think, I hope you don't get bored. But I would like to frame this in the perspective of being an early career researcher. So I will tell you a bit about my path. So Priscilla who? I have my undergrad, I am Mexican. I did it back at home in Mexico. It was in chemical biology. And before then, I always thought microbes were pathogens. And that's, I think, like a common misconception. And then it was then that I decided that chemistry was interesting, but that microbes were the best chemists there are. So I focus on microbiology as the production of chemicals from them. And after that, I then work in an industrial R&D lab because I decided I wanted to do research. But during this time, I noticed that was not for me. I wanted to go back to academia, which led me to apply and to get a grant to study in the United Kingdom, where I did a master's degree in environmental engineering, which maybe sounds, most of you might know, it's mostly environmental microbiology, but applied. And then I did also a PhD there and was in environmental biotechnology, where I tried to convert municipal solid waste into bioethanol. And so, but then this happened, and it was thanks also to another microbiology society, one from the United Kingdom. Now it's called Applied Microbiology International. And well, this is what happened. It's not a flex, but in case you don't know, this is Sir David Attenborough. And in that meeting, he had just released his the second issue of Blue Planet 2. And in this documentary, he talks a lot about ocean pollution by plastic. So I decided, well, I know microbes can transform pollutants. Why don't I work on that? And so I moved from municipal solid waste, like organic waste, food waste, and bioethanol, and I expanded that to plastic pollution. And since then, I've been doing research in plastic pollution, microbiology or not related. But then, of course, you know, the pandemic happened, and I have this thing that microbiology, molecular biology, gives you tools to be of use to society. And so I work in the National Health Service in the United Kingdom as well. And in this picture, you can see we are posing proudly because we had processed over 2,000 samples of patients in the lab. But that also meant that each sample came in a plastic tube, right? And for each plastic tube, there were multiple pipette tips used, and all of them going to the bin. And every time, clean gloves, and every time packaging of these materials. And because this is like, of course, dangerous samples, then you might expect that that this is just incinerated, right? It's single use. And I was overwhelmed. We were doing something good for health, but at the same time, we were consuming and discarding so much plastic. And that is when I started thinking of this perspective. Not quite yet planning to write it, but I was overwhelmed by how much plastic we use. Then I moved to Switzerland, where I did a postdoc, mostly on the plastisphere, the microbes that get attached to plastics. And I am doing now another postdoc, but this is in the University of Zurich, but it's more regarding the perspective I wrote for FEMS, integrating sustainability practices into the labs where I work. And with that, I want to tell you then, what is the overarching topic of my research? It looks like I've been just jumping here and there, but really the topic is waste. First, valorization, like with ethanol, identification of microplastics in marine environments, monitoring also in marine environments, and now elimination, mostly in research labs. So I could say I'm like this guy. I love waste, but mostly I see it as a resource. So from that perspective, that's how I see plastic. I think we can use it or eliminate it. Finally, I also, before I move into the publication, I wanted to talk about my career in terms of FEMS. So when I did my PhD, I got also a meeting attendance grant. My first conference was a FEMS conference in Valencia. Then I also got a research and training grant. I did 3 months internship in Chalmers, University of Technology, that's in Sweden. Then I also applied and was successful in obtaining the summer school for postdocs in 2022. This experience really changed my career, I think, my view of science, and I felt more confident after it. And I think it opened many doors. So, and finally, I submitted this manuscript that now I'm presenting, the perspective, and I just submitted an original research article, but that's for another presentation, hopefully. So with this, I just want to tell you, I have asked other, professors that I very much admire, what is like the secret, not secret, but what do they recommend for like an academic career? And they all talk about the impact of having a mentor. And I have had very good bosses, very good supervisors, but I don't think I have had a mentor, somebody that would introduce me to people or connect me to networks or tell me like, you know, not secrets of the trade, but guide you in the right direction. And I would say FEMS had somehow fulfilled that function by networking, by connecting with different people here. So all of this, when I'm talking about these grants, what I want to tell you is apply for them. You already, you don't miss anything by trying. And then you can meet new people, and sometimes you form connections that will last for many, many years, and I hope for a lifetime. So, also I'm part of the committee member of the upcoming Congress, so I hope you will join us. But yeah, this sounds like an advertisement. But with that in mind, I now pass to the perspective I wrote. As I said, it's different. It's not original research. It's a perspective from plastic pollution, motivated by my work on plastic pollution. So this is it. This is the perspective that got published in 2024. Clean your own house first, integrating sustainability into microbiology labs. And then first I want to talk about, again, plastic pollution. It is everywhere, as you know. So in this paper, for example, I am a co-author. we found polyethylene terephthalate fibers inside of this species. That was the paper. Actually, if you see it, it doesn't have plastic on the name, because it was mostly about the identification of a new species in the Mariana Trench, which is the deepest environment on Earth. However, it was new for humans to meet this little crustacean, and it already had plastic inside. So we have found plastics there at the bottom of the ocean, at the top of the mountains in Everest. We have seen them in every sample of drinking water that has been sampled, and look for microplastics. There are microplastics. We have also found them in human poop. We have recently found them in placenta samples. Also, most recently in brains of, and most tragically, in all samples of German beer. So we know we have a problem, right? I don't want to scare you. There are many other pollutants everywhere, but so plastic are really a problem. And you might think like it's just something societal and bigger. But then we can think about our own impact as researchers, because I think many of us do research that is focused on the environment, on solving environmental problems. But at the same time, our own research generates waste, like single-use plastics. For example, academic research worldwide contributes to around 2% to the global plastic waste problem. That is a lot. And that's approximate to 5.5 million tons of lab plastic waste. And in 2000, this is a figure that was calculated in 2014, so over 10 years ago. Probably now we have more. And that was just an estimate done by one university in the UK. Then we don't have a specific number or an approximation for Europe. I put Switzerland because that's where I work, but you can substitute Switzerland for any country where you are working. And then your institute where you work, where I work, probably we produce around 5.7 million of 2 liters of empty plastic bottles. So that is a lot of plastic. And again, this number comes from 2014. I don't think we have done much to decrease that number generally, so probably it's more. And then that makes you feel like we can do something, we should do something about it. So what we can do? I will just tell you very briefly what I'm doing now in this project I'm working on. Well, it's called Deep Wash Project. We have this machine. This is not an advertisement. This is a machine. The producer is in the United States, and it is designed to wash pipette tips. So we have to validate, of course, that the pipette tips actually get properly washed, that whatever they are still structurally safe to use, like you are still pipetting the same volume, that there's nothing attached. And in Switzerland, at least, we also have to prove that what we are discarding into the sewage is safe. So that's also something we're trying to prove to the authorities. But if we manage and then convince the other team members of the lab that this is okay, then we can save also money apart from resources. But this is just one of the measures you can take. There are other ways to reduce plastic waste. For example, I mean, this is not new to microbiology labs. Go back to using glass and metal materials, for example, glass Petri dishes, which is what I do when I now that I'm working again with culturing. We can use glass L-shaped spreaders. We have seen now we substitute those for single-use blue plastic rods, right? Metal inoculating hoops. We also have seen how that has been substituted by plastic ones. I still don't know why. Then another action that we can take is develop and use protocols for reusing plastic consumables. For example, there are already methods available that were published some years ago, proving that tips and microcentrifuge tubes are safe to reuse, even for samples that had COVID on them. So that's quite impressive. And this not only address environmental issues, but the institute that proved this, the author is there also. If you read my paper, you have all the references. They also had this problem of the shortage of materials during the pandemic, so they could not find tips. and then they develop this method. So you are addressing economic, sustainability, and availability issues. And another thing that you can do is plan, buy, and use better these materials. What do I mean by plan? Think about the waste to be generated before starting an experiment. I know you are mostly thinking of like, okay, I want to get a good experiment done, but you can include some sustainability considerations when possible. Excuse me. So, and then, for example, just are you gonna use the full 19.6 well plate, or can you share it with other colleagues? Or there are also plates that have, I don't know what's happening to my voice, sorry. Okay, and you can also engage with suppliers and waste management companies. In the article, I mentioned some examples where some labs have engaged with some manufacturers, and now they are using recyclable materials for their tubes, or they segregate, wash, and treat differently some materials that can be reused. Another thing that we can do, of course, is not everything is about plastic waste. Sorry, that was. There are many other actions that can be taken. Probably some of you might be aware, there's this freezer challenge where it has been proven there's no need to have -80 degrees to store long-term samples. And there's even like a challenge where you can put like, you can register for this. In the paper, there's also the reference. It's an initiative by a lab in the United States. And then by that, you can really save money and energy use. Also, you can just starting with simple measures like switching off equipment when it's not in use, programming batch use of equipment that you can plan with your colleagues. And then there's also a toolkit developed already that you can follow, and you can kind of do an audit of your own lab. And I think this can be a fun activity that you can do with your lab group. Another thing that we can do is also the chemical use and disposal, a proper inventory management. I don't know how it is in your countries. In some of the labs I have worked, sometimes each project has their reagents. So if my project has a different funding, then I will have my own set of reagents and a different one, and so on and so on. Some places share everything, so I'm not saying this is the norm, but it just makes sense to share materials, because at the end my project will end and those materials will get there, and they will next person will have their new and blah, blah. So having an inventory management and sharing materials, it just makes sense. Also eliminating or changing the hazardous waste that we use is a way to saving in not only waste, but the treatment, because then we can just discard it without having to go through a different route. And centralizing the use and disposal as well. And maybe your institution at bigger levels can help you with that. Another one, and I think maybe the most important, is education and awareness. Try to raise culture of environmental awareness, because sometimes you have members that are resistant to that. To be fair, sometimes we do research for 40 hours in a week or so, and you're thinking like, oh, do I have to bother about that? Do I have to put the tips in this other box to get washed? And I know, I understand it is tiring sometimes, but by having open communications and regular training and discussions on sustainability, I think you can get your group on board. And as I was saying, lab sustainability is a teamwork. It doesn't mean that you as an early career researcher or as an individual, you will change the entire system, but you need to have a team. For example, you can ask your institution, university level, department level, maybe there are already some measurements in place for sustainability. Mostly, well, from what I have seen, is mostly about waste. I don't know, some other places have more, some others have almost nothing. If there is one, engage with your sustainability office, and you can learn many initiatives that they have already in place, and maybe you can convince your boss or your group to join. And if none of this is in place, communicate the importance of sustainability to your representatives. Maybe your student representative, your postdoc representative, some higher up that maybe can amplify your concerns. Learn societies. Join training and other opportunities that they learn societies, I mean like microbiology societies. I've been part of the sustainability member committee of Microbiology Society of the UK. I also worked in FEMS, of course, and in other different events. So whenever I see something related, I join because it's something that is very important to me. Also, there are journals requesting sustainability measures. Maybe you could think of submitting your work to them. There are grants available where they can support maybe your training on that or some activities in your group. And if not, write to the editors or write to the committee of the society about your concerns. I think societies, in my experience, are very open to listen. And as the topic is early career researchers, right? I would say that is our job, but also of all the team to make a difference. From many of the references I cite in my paper were led by early career researchers. PhD students, postdocs, doesn't mean that, but then I remember in the review process of this manuscript, they told me, the reviewers, well, it cannot be only the early career researchers because the nature of our contracts is temporal, right? We leave the workplace and then will these measures still be there? So you definitely need to engage also the professor, the technical team, so that this is a long-lived process. And okay, initiate discussions if not already in place, present the alternatives, and what are the opportunities for saving not just energy and environmental concerns, but also money. I think everybody wants to save a bit of money, especially if you are the manager. And ask for support and advice. Probably the technical team also knows some, can make very valuable suggestions. So with that in mind, I just want to, this was my, like a summary of what I just spoke. There's far more resources and activities that you could do to improve and integrate sustainability into your lab. But I just picked some because I didn't want to make this too long. And as I said, you will not only, by doing this, you will not only help the environment, but you will always also save money and increase the efficiency of your lab. And then, so about lab sustainability, if you have any other ideas, let's talk. I would really like to hear. If you don't have questions, maybe suggestions are also welcome. Thank you very much. I will stop sharing now.
Okay, thank you, Priscilla. Very good stuff. A lot of great information there. Hopefully my brain continued on because you really caught me at microbes are the best chemists. And but okay, now Max, shall we begin answering some of our many questions?
Yes, definitely. Yeah, we can get everybody back on screen as well. And so thank you all. Really wonderful talks and enjoyed very much. And again, very different topics, which I think it's also a lot of fun to go in. Before we get into the science, I noticed there are a number of questions about joining a society. How do you do that? And actually, how do I join FEMS? Which is actually a bit difficult because FEMS is a federation of societies. So in order to be part of the sort of FEMS family, you actually join one of the many societies that are part of the federation. And that would be sort of the way to go about. You can also be what's called a FEMS affiliate. And that is just sort of a connection directly to FEMS to get information by email or so on in terms of how to do this. So again, take a look into the link on this. But again, we're not directly members of FEMS, but we become part of the FEMS community through the various societies that we belong to. And you could join many of them that actually also belong to FEMS.
Yeah, so for example, I'm in Germany, and so I joined the German Society for Microbiology, and that links me into FEMS, so I can apply for all the cool grants and, like, you know, get cheaper registration, et cetera, et cetera.
But equally, I want to plug in, I mean, other societies in the United States and America. There's the American Society for Microbiology. There's the International Society for Microbial Ecology, and so on and so on. So there are many, many different ways to become active. And was there anything else on these? Okay, yes. And I know that, yeah, Jessica and Juan, you've already answered a couple of the questions that have come in, but there were a few that I thought would be interesting to go back to, and that's just sort of this question on methane consumption in different environments. And again, there were questions both on the impact of nitrate, but also other ecosystems. And maybe we can get back to that, and maybe this aspect about aerobic versus anaerobic methane oxidation. I don't know, do you want to jump into that and how that could also be a factor also in the system that you studied?
Yeah, thank you. Yeah, I think those are very important and interesting questions indeed, because it makes also the extrapolation of, or the prediction of methane emissions more difficult because it changes so fast, or because it's so variable. And yeah, I'm a bit lost on where I should start, because it's like now it was a very big question.
Well, I think one of the things you pointed out was how different electron acceptors, the presence and absence of sulfate or presence and absence of nitrate is key.
Yeah, indeed. So depending on the availability of these, you get either more methane produced, because if the electron acceptors are not available, then there is less organic matter that is respired, there is more that is fermented, which then will lead to methanogenesis. And on the other hand, you also have more or less, less methane can be oxidized again, because you are missing the electron acceptors for this process as well. And I think there was also another question on how you could mitigate these methane emissions. And I think one of the main points is really, either add electron acceptors and also reduce the input of organic matter. So reduce eutrophication, reduce the input of nutrients into aquatic ecosystems, so that there is not that much organic matter production in the first place, or there's not that much organic matter input in the first place. And then I think this is also, I find the topic on oxygenation of water columns very interesting. So there is lakes and other systems that are being they add oxygen there, they bubble it with oxygen to add this or to prevent these things from happening. But I think this really depends on the system of how well this will work, and if it doesn't then disturb more other natural processes that are happening, which is the stratification and so on. And I think these techniques are very, very valid, but I find it interesting to think about this as well.
Now and then when you move on also to soil systems, rice paddies or so on, there's even more interconnections that are of interest. Yeah. Let's jump to, again, something completely different, beetles and their microbiomes. There was a question about how they're acquired. So is it vertical or horizontal transmission? And actually, regarding just also the microbiome that you studied, were you looking mainly at the surface and the cuticle microbiome, or these are gut?
Yeah, exactly. Well,
combination.
there's a lot of evidence that they have a vertically transmitted microbiota. Actually, when we talk about the fungi that are associated to them, we know that they can transport in specialized organs that actually produce certain enzymes and substances that are antibiotic and kind of select specific microorganisms. And we know that these mutualist fungi are transmitted from the mothers to their offspring, that are usually females, because these beetles in many cases are parthenogenic, or they just reproduce with their brothers. In the case of the bacteria, we know less about that. Previous studies about metabarcoding have tell us that the bacterial community associated to different species of ambrosia beetles is very species specific, and that probably this is making that they are vertically transmitted, because even beetles that cohabit the same environment have different communities. There's another question there actually about why there's a kind of an effect of selection, or how is the effect selected in the antimicrobial effect. And actually it's something related to that too, because I think that probably this antimicrobial effect that the Erwinia has is given to coevolution. Actually, probably this bacteria, that's my hypothesis, but probably this bacteria has been evolving with the beetles somehow, because they interact together. And certain microorganisms that usually interact with the beetles are kind of affected by this interaction. For example, some species of Trichoderma, actually I didn't mention that, but I tested species of Trichoderma that are used as commercial products in biological control, and I got better effect in the species that are naturally occurring in the fungal galleries of ambrosia beetles. And that's the same for the entomopathogens. Beauveria bassiana is usually found in ambrosia beetle galleries, and it's having a negative effect, the Erwinia, in this fungi. And actually, related to the topic, where can we find this bacteria, is kind of complicated, because the studies that we have to date in metabarcoding, well, we only have studies on the wall beetles. We don't have anything no information about the microorganisms inhabiting the gut, for example. We have some studies on the fungal gardens, and actually I isolated bacteria for my studies from the fungal gardens. So we know better actually the community composition of the fungal garden than the community composition of the beetles, because in general we don't know about compartmentalization in the beetle and where the microorganisms are located.
Okay, no, thanks. Let me jump to plastics. So I saw one question on there, and this linked to water. And again, and this is sort of the question of how to actually calculate the environmental cost, because water is also expensive. I mean, the treatment of water, the treatment of wastewater. So the aspect of how much do we actually consume energy by washing? And have you thought of that, and how to get those numbers to actually compare to, let's say, is it better to just discard a pipette tip?
Yeah, that's a good question. And it's actually something I have to prove to convince the group that using this machine is good. So we are collaborating also with the energy team of the university. We are going to do a life cycle assessment, but you have to then also consider the production process of a pipette tip, right? It's like from the fossil fuels extraction, the use, the conversion, all of that. And at least this machine uses maybe like 10 liters of water per wash, but then you have like pipette tips that you can reuse. And there are other methods. Of course, you don't need this machine. In many other countries, I remember in Mexico when I did my undergrad, we were washing our tips and autoclaving them. more, not so much for environmental concerns, but because you don't want to spend so much money on using tips all the time, like, and wasting them. So in that sense, we expect that it will be significantly less to reuse them, but we are working on having specific numbers, and we're going to have the energy calculations of energy use and also water use, and then have a bigger, better picture of that. But yeah, it is a valid question, of course.
Well, I started my career just as plastic tips were becoming more abundant, and so most of us used actually glass pipettes that were washed and then re-autoclaved again. So maybe we'll go back to glass even.
Yeah, but I have a question. Could you measure very small quantities with a glass pipette?
No, of course not. No, I mean, and that's of course the aspect of being able to reproducibly add microliter amounts is of course what is the huge advantage. But no, there's definitely, I mean, thinking of all the energy consumption, the freezers, I mean, do you need minus 80 degrees or is minus 65 equally good for your cultures or so on? I think that's another aspect, and sort of thinking of these indirect costs of what we do in the laboratory as well. Okay, are there other questions that you've seen in terms of the Q&A that we want to get into? I'm trying to sort through.
Maybe one question that I see that might be interesting for Jessica also. Looking into the role of sunlight into the community of methanotrophs, do you think it might have any impact on the efficiency of the biofilter, methane biofilter?
Sorry, the impact on what?
Sunlight.
Sunlight. Yeah, good question. There is a study that looks into how sunlight might impact methanotrophs, and it has been postulated that sunlight can inhibit methanotrophic activity. And it's also, like, it's controversially discussed why there is so much oxygen in the top water, and also there's still some methane, but then the methanotrophs are not there actually. So it has been postulated that this has something to do with light inhibition. Other studies show that they are not inhibited by light, so it seems to be maybe dependent on which methanotrophs it is, or it is in connection to phototrophic activity or other things that this is affecting it. Yeah.
But indeed, in your result, it looked like we saw the much higher abundance of methanotrophs in a deeper water column rather than the closer to the surface, right? So it kind of could indicate that there is some kind of link.
Yeah, indeed. We cannot exclude it, but I can also not confirm it. For our study, we didn't really check it. But I think that also other like that every microbial community and functional group has its own niche in the water column as well. So it might be that the heterotrophs and phototrophs dominate the top water, while the methanotrophs find then their niche in the lower oxygen conditions where they are more competitive compared to in the oxic water or in the top water.
Makes sense.
I
I don't want to. Oh, sorry. You go, Bea.
Oh, thank you. Sorry, Damien. I can jump in and ask a question for Juan Carlos. There is a question regarding which antibiotics produced by Erwiniaceae that I know you didn't target that question in your paper, but like they talk about how they inhibit one fungi, but seems to not inhibit another. And one of the things that I noticed from your presentation, from your paper, is like it's really species specific. So we cannot like generalize by genus. So can you give us a bit of your intuition about all of these complex interactions between the microbe communities that you have seen and why it's important probably to do these essays.
Yeah, thank you, Beth. Actually, I think it's related to this specificity that we see in the effect of the antimicrobial effect in these different fungi is related to the co-evolution, actually. In some cases, I tried fungi that are used as commercial products, for example, some certain species of trichoderma and certain species of entomopathogens. And I also tried isolates that I got from my microbial collection on the field in the galleries of ambrosia beetles. So when I got the isolates from the galleries, in most of the cases, I have an effect by the bacteria. And when I tried the commercial products were not affected by the bacteria. So my idea is that probably microorganisms that are naturally interacting with these bacteria and with ambrosia beetles are already inhibited by the bacteria because they have been evolving together for many times probably. And that's not something new. Actually, we know that from other bacterial, fungal, insect interactions, for example, in leafcutter ants or termites, that is very similar.
There's a follow-up question that actually just came up here, and that's about the nature of the antibiotics. So do you know actually what the chemical is that Erwinia is producing?
I don't know in detail because I didn't describe the molecules, but I know there are some polyketids produced by the bacteria.
Okay.
There's also some small proteins probably. There's also, yeah, kind of, but they are not described. And actually, when you compare the clusters to clusters that are already described, not all of them. They don't have 100% of similarity. So probably they might be new molecules in some cases that we don't know about them.
Okay, maybe well, we've probably need to start wrapping up here. It's been a wonderful 1.5 hour discussion with everyone. And first, I want to, of course, thank everybody here in the audience. It's actually from the names I've seen on here, it's worldwide. And that's actually one of the exciting things is that we can really reach all over the world with these webinars. And then, of course, thank the FEMS and Oxford University Press staff for a lot of work that happens behind the scenes for these webinars to happen and run so smoothly. So thank you. And then, of course, a great thank you to our hosts and speakers, Jessica, Juan Carlos, Priscilla, Martina, Damien, and Bea. So it's been wonderful, and I hope to see many of you in the audience at the FEMS Congress in July. So thank you again for taking part in this and all the work, and for publishing in FEMS Microbiology Ecology, of course.
All right. Bye-bye, everyone.
Thank you.
Bye-bye.
Thanks. Bye.